Hi, everybody. Hopefully everybody can hear me now. It's a big day in science today, not just because you're going to listen to Erin Vanzyl right now, but today the Nobel Prize in Chemistry was announced for work developing the CRISPR system, and two female scientists were awarded the award today, and that's a first. So exciting day. I'm happy to be hosting here and introducing Erin Vanzyl. She is a graduate student in my lab. She's working towards her PhD currently, and we'll be talking about the background behind her work and a little bit of her work at the end of her talk. She's been at Carleton now for quite a few years. She's worked on a number of different projects, mostly in my laboratory over the last few years, and so I'm really privileged not only to be supervising her, but to be introducing her today to let her talk to you about something called pre-mRNA splicing, which she has called the splice of life for her talk today. So please welcome Erin.
Okay, so hopefully everyone can hear me, and I'm going to go ahead here and share my screen. So maybe Bruce, if you can give me a thumbs up. Are you seeing my presentation here now? Perfect. So thank you, Bruce, for the introduction. And as mentioned, my name's Erin, and today I'm going to be talking to you about my work, so gene processing, the splice of life. To start off with, I'll be giving some background on the flow of genetic information, as well as what genetic information actually is. And the flow of genetic information starts off with DNA, or deoxyribonucleic acid. And I like to think of DNA as the blueprints of an individual. So DNA is what makes up an individual's genetic code, and almost everything needed to make up a person or an organism is in their DNA. And as you can imagine, this is a lot of information to be stored within us, taking up very little space. And to highlight this, each cell contains 2 meters of DNA stretched out end to end. And we have 37 trillion cells, which means that a single person's DNA could be stretched from the sun or the Earth to the sun and back 61 times, which is completely amazing. And if you take nothing else from this presentation, at least remember that fun fact. So when a cell divides, this DNA must be copied, and this is done through a process called DNA replication. So the DNA in each cell is made up of billions of very small individual building blocks, which are called nucleotides, and these must be replicated and stay in their sequential order. And each day, 2 trillion cells in our body divide. And now that we've covered what DNA is, we can talk about what a gene is. And a gene is simply an area of DNA that has the ability to encode a protein. So if this is your DNA, your gene is just a specific segment of that DNA that has the ability to encode proteins. And now moving on into what a protein actually is. A protein is a functional unit of genetic information. So proteins carry out a wide variety of functions within cells. Some examples being keratins, which form your hair and nails, enzymes, which break down your food, and antibodies, which function as part of your immune system. So now that we know what DNA is and what proteins are, we can move on into how these relate to one another. So ultimately, DNA aids in the formation and encodes protein production. However, there is an intermediate that's very important in this process and is needed for DNA to encode a protein. However, for simplicity and to build some suspense, I'm going to keep this as a black box. And the important takeaway here is that the flow of genetic information goes from DNA to protein with an intermediate in between. And now that we know what DNA is and what proteins are, we can connect the two and talk about what happens when there's an error in DNA and how this can affect proteins. So sometimes an error in your DNA can occur, and if this error becomes permanent, it's called a mutation. So DNA errors or changes, which are mutations, can therefore influence the protein that's being formed by this particular gene. So looking at the bottom here, if we have a cell's DNA and this has been replicated, and now the DNA is acquired an error or a mutation, this will be passed on into subsequent cells. And some of these mutations can cause changes in proteins that are linked to genetic diseases. So for example, again, up here we have our DNA, which is encoding a protein. And if we have a mutation in our DNA, which changes the protein, sometimes these altered proteins that are caused by DNA mutations or changes can be linked to genetic diseases such as cystic fibrosis, Alzheimer's, and cancer. And today I'm going to be focusing on the flow of genetic information and how it relates to cancer specifically. So to start off, what is cancer? Cancer refers to a series of diseases with the shared characteristics of cells with the ability to grow uncontrollably and have the potential to spread to other parts of the body. So at the top here, we have our DNA, which is helping to make a protein, and we have a normal healthy cell that will divide a certain number of times and then undergo cell death. However, if we get a DNA mutation that then alters the protein, we can now get a mutated cell with the ability to divide uncontrollably and avoid cell death, which can give rise to cancer. And there are two general types of mutations that can lead to cancer, and these are either loss of function mutations or gain of function mutations. So both types of these mutations cause cancer, can cause cancer. The first is from shutting off the expression of certain genes, and the second is from turning on the expression of certain genes. So looking at these each individually, starting with gain of function mutations, these are too much of a good thing mutations. So this is when good gene function is exaggerated, which can be bad. For example, genes that drive a cell's life cycle are good. However, too much drive may prevent this cell from ever undergoing cell death, and that can be bad. We also have loss of function mutations, which is when good gene function gets turned off. So loss of function mutations that lead to cancer commonly occur in a class of genes called tumor suppressor genes. These genes, the best studied example being the p53 tumor suppressor, are expressed in cells and help to prevent uncontrollable cell growth and the formation of cancer cells. However, sometimes a mutation can occur which prevents this good, helpful protein from functioning. And below, I'm just kind of comparing it to your smoke alarm, which would be your tumor suppressor. And that's a good thing, and that's helpful. However, if you have a mutation, or say the batteries die in your smoke detector, that is now a problem. And now going back to our flow of genetic information, where we have DNA to protein, and the moment I'm sure you've all been waiting for is what is behind this black box? What is our missing link between DNA and protein? And the answer is RNA. So the missing link here is RNA, and now we have a simplified version of the flow of genetic information. So we have DNA, which can encode RNA, which in turn encodes a protein. So what is RNA? RNA is ribonucleic acid, and it's the intermediate between DNA and proteins. So DNA is in a particular area of the cell known as the nucleus, and in the nucleus, the DNA makes a usable copy of itself that can be shipped away into the cell where proteins are made, and RNA is this usable copy. So DNA, which contains two strands wound together, is opened up, and a single-stranded copy is made of a portion of this DNA, and that is RNA. And this process is called transcription. And in the end, the DNA is annealed and left intact in the nucleus, and the RNA is shipped out and able to go out into the cell and make proteins. So going back to my DNA is the blueprint, or your blueprints analogy, then your RNA is a carbon copy of these prints, and it can leave the office and be used on site for building whatever the blueprints are for, which in this case would be a protein. So now we have our RNA copy, and it's been moved or exported from the nucleus. And in the cases where proteins are being made, this RNA is now bound by little protein makers, which are called ribosomes. And these ribosomes use the RNA as a template to encode a protein strand. And this is done through a process called translation. And these little ribosomes or protein makers are in a specific part of the cell, or often found in a particular part of the cell called the endoplasmic reticulum. So all of this taken together, the flow of genetic information for DNA, which makes a copy of itself called RNA, which makes a protein copy, is termed the central dogma of biology and was initially published in 1958. And this model appears very linear in that you have your DNA, which will always give rise to the same single RNA, which will always give rise to one protein. However, it's actually much more complex than that. So to start off with, not all RNAs are made into proteins. However, today I'm going to be focusing on RNAs that are used to encode proteins. And RNAs that are used to encode proteins contain two different regions. So to start off with, you have exons, which are these yellow boxes, and introns, which are the purple line. And before this RNA can be translated or used to make a protein, these introns have to be removed and the exons have to be ligated. So we have something like the bottom here with all of our exons together and our introns have been removed. And this is done through a process called pre-mRNA splicing or just splicing. And so I have an example here at the top of an RNA that is going to be translated into a protein. And RNAs that are translated into proteins are called messenger RNAs or mRNAs. So this messenger RNA needs to have its introns removed, which is again done through the process called splicing. So I've used a letter here to label each individual exon. So we have exon B, E, A, and D. Each of these exons need to be joined in a specific sequential order to give rise to the correct protein, or in this case, the correct word. So let's say all introns are removed and all exons are ligated. We would get exons B, E, A, D, which makes the bead protein. However, sometimes an exon can get skipped or any variety of errors or alternative events can occur. So as seen here, if we skip the A exon, then we get exons B, E, D, and now we get formation of the bed protein. Similarly, if E gets skipped, we get the joining of B, A, D, or the formation of the bad protein. And this allows for there to be variety in the protein formed from a single RNA. Sometimes this variety can be a good thing. However, there are cases where these alternative proteins, or in this case, these bad proteins, can give rise to diseases such as cancer. Many events like this, which are called alternative splicing events, have been linked to the progression of cancer cells. Therefore, inhibiting this splicing process is being investigated as a possible cancer therapeutic. And this brings me to my research, which looks at the consequences of inhibiting splicing in human cells. So again, splicing is the term used to describe the process of removing these unnecessary introns and joining these exons. And this is done through a long, complicated process carried out by the spliceosome, which I'm just showing here so you can appreciate the complexity. However, the important part is really this: introns removed, exons joined. So in order for me to study what happens when splicing is inhibited, I need a way of inhibiting splicing. And to do this, I use a compound called isogengetin. And isogengetin is derived from ginkgo biloba trees. It has anti-tumor properties, as well as the ability to inhibit splicing. So one of the first things that we did in the lab was complete a gene analysis of isogengetin treated cells. So here we were able to take cells that we had treated with this splicing inhibitor as well as untreated cells, and compare the expression of almost every gene in the human genome. This amazing technique allowed us to get a general understanding of what genes and what pathways seem to be altered when human cells are treated with isogen-getin and splicing is inhibited. We found that the inhibition of splicing seems to activate a particular stress pathway, specifically an endoplasmic reticulum stress pathway. And if you remember, the endoplasmic reticulum is the area of the cell where a lot of these protein makers or ribosomes are often found. Many proteins are made in the endoplasmic reticulum, and at this point we had information indicating to us that when we inhibited splicing using isogen-getin, these cells were activating an ER stress pathway. So how can we confirm and investigate this? To first determine if splicing inhibition was causing this cellular ER stress, we need to know the characteristics that are associated with the stress response. And the characteristics associated with the stress response can be put into two groups. So first, there's an increase in helpful proteins or genes, and second, there's a decrease in overall protein production. And this may seem conflicting, but from the cell's point of view, the situation is something like this. When you realize you've left the tap running and there's water overflowing, you do two things. First, you turn off the tap, and second, you go grab some towels and a mop. In this situation, the towels and the mop are the increase in helpful proteins. These things help alleviate the stress that the cell and the person who overflowed the sink are feeling. Turning off the sink is to not continue to add to the water overflow is the equivalent of stopping protein production. Since the endoplasmic reticulum is the site of protein production, it should not continue to form proteins when it's not working properly. So back to the lab setting, we've used a variety of techniques, and lucky for me, some of these have worked to measure these helpful genes and proteins, and this is shown on the right. So DDIT3, DDIT4, and SHAC1, the names aren't specifically important, but these are all helpful genes or proteins that get activated when we have ER stress as part of the ER stress response. And so ND corresponds to my no drug or untreated controls, and IgG stands for isogen-getin. And the main takeaway here is that each of these three targets increases significantly when we treat with isogen-getin, which supports the idea that we are causing this ER stress response. This increase is helpful, this increase in helpful genes and proteins indicates again that we are activating this pathway. So now we can look at the other group of characteristics, or the other key characteristic, which is the inhibition of overall protein production. To do this, we used human cells that have been modified to express a very short-lived fluorescent marker called green fluorescent protein, or GFP. So these cells require constant formation or translation of GFP in order for the green to remain visible. Therefore, we hypothesized that if splicing inhibition were to decrease protein production, which would indicate ER stress, we would be able to see this by a decrease in the green fluorescent protein being expressed in these cells, which is what we can see on the left. So again, ND and DMSO are two controls, and these cells are expressing the green fluorescent protein. However, after we inhibit splicing using isogen-getin, we get a large decrease in the visible green protein, and this supports the idea that we're getting a decrease in overall protein production through this ER stress pathway. And this brings me to where I am now and my future directions. I will continue to determine the key genes and proteins involved in this stress response. Specifically, I can use CRISPR-Cas9 technology to target specific components of this pathway that we believe to be important and study these effects. And we were so excited we had to tell you this twice, but science in the news. So yeah, the Nobel Prize was awarded to 2 researchers in chemistry this morning for their work on developing the CRISPR-Cas9 editing tool. And I'm fortunate enough to be able to use this technology and this technique in my lab to help aid in my project. And overall, these experiments will help us understand the fundamental processes that are involved, as well as provide rationale for possible treatments to cancer. And I'd just like to thank my supervisor, Dr. Mackay, as well as Victoria, Alex, and Hadil, who all worked on this project, and all lab members, past and present. And with that, thank you everyone for listening to me talk about my research. I know I think it's super interesting, and I hope you all did too, and I'm happy to take any questions. And I guess I will stop my screen share. And I don't know if we have any questions.
No questions as of yet. Feel free to type into the Q&A section. Oh, here we go. Can you read those, Erin, or do you want me to convey the? So we have a. Sorry, go ahead.
No, yeah, I have. I can read the Q&A from my good friend Anisha. She said some very nice things. So there's another question. It sounds as if inhibiting splicing would cause more problems than it solves. Good observation. So yeah, obviously inhibiting essential processes isn't a good thing to do. However, in terms of the applications of this being to possibly use it to treat cancer, it causing harm is not a bad thing. And cancer cells specifically seem to be more susceptible to splicing inhibition than normal cells. And not to get like too into it, but it seems that using splicing inhibition in combination with other chemotherapies seems to make them extra susceptible. And in this case, that's a good thing. Is there any way to specifically target the splicing of oncogenic cells at this point? That's a great question. I don't specifically know if there's a, I mean, I guess, I can think about this. Not that I know of off the top of my head. You could target, I mean, you could use, I assume, so many technologies to edit oncogenic cells and sequences, but I don't know if there's a way to specifically inhibit their splicing. And the splicing inhibitors I use are general splicing inhibitors. If that answers the questions.
Any other questions out there? I suppose somebody could devise strategies to target important known single splicing events in cancer. Yeah, but not the more broad fundamental underlying errors in splicing, I guess. Anyone else out there? Anyone else want to ask anything about CRISPR? About the news today?
Oh, there's one more.
There we go.
So what would treatment look like and how would this be administered into treating a patient? I'm glad because I briefly thought, I bet they'll ask me this and I should quickly look at it because this isn't quite my realm. But the splicing inhibitors that have been used in clinical trials are intravenous. And I believe they were also like given with the chemotherapeutic treatment. And that's still very early stages of research for that.
There's another one there, if you can see it.
Oh, sorry, I have to scroll down.
Oh, they're coming in quickly now.
Yeah, okay. What is one tool or technology that can make your research easier? I mean, if I could pick one that I could just choose to make work, that would be Western blots, but they they don't. So a substitute that makes Western blots work would be great. So I did hear that CRISPR-Cas9 can introduce serious side effects. Any thoughts on that? If in side effects mean as if we're actually doing it in organisms, I don't doubt that it can have tremendous off-target effects. It can also do that just on a cell basis. For how I'll be using it specifically, it's on dishes of grown cells. So they're definitely maybe like off-target effects in that case, not overly side effects if we're not counting it as an organism. Hopefully that kind of answers that if we're just looking for my thoughts on it. So what types of cancer have been used in this clinical trials? Catherine's asking me that. I mean, Catherine, I'll send you the paper if you want to read it. I can't remember exactly what type of cancer it was. Bruce might know.
I think
you know.
Certainly at the preclinical level, it's multiple cancers.
Okay, I've only been super familiar with the one paper. Thank you for the presentation. Thank you for listening. So how do researchers such as yourself become aware of all the techniques to use? And then when you do get recipes, how do you master them and how do they work? I like that. So I become aware of the techniques mainly because Bruce here helps me and teaches me techniques and we go through them in the lab. Especially at a lower level, it's a lot of kind of being guided towards what techniques to use. And a lot of labs will use very similar techniques. And then, you know, I still try to Google all the time different splicing assays and keep up with literature on what cool things people have done. So then when you do get recipes, how do you master them and know that they work? So you often have a lot of controls and so you have different like going back, if you saw my example of those 3 targets that were increased, to know if that target was accurate, I get something that I know will increase those targets and compare it to those. So you do a lot of positive and negative controls to know that they work. Also, in terms of a Western blot, you look at it and it's either going to be beautiful or it's going to be horrible, and you know right away if it's worked. And in terms of mastering them, I don't know, I've been in the lab for 6 years and I still definitely haven't mastered all of them, but hopefully, eventually practice will make perfect. Awesome. So Catherine wants the paper and promising area of research. Thank you.
Anyone else? No.
No.
Okay. So thank you very much for taking the time to come and hear about work being done in the faculty of science and the Department of Biology. We're happy to host and hope you'll come back and participate in any other seminars in this series. I think we have one next week.
Yeah.
Yep.
I think it's a COVID related one next week, so very relevant.
COVID vaccines, I think.
Okay. Awesome.
Fantastic.
Okay.
Thank you very much to you too, Erin.
Oh yeah, thank you everyone for listening to me talk.
Okay.